So, very good evening to the students who have joined today's live session. We will wait for a couple of minutes and then we will start today's live interaction sessions for this NPTEL course of Organic Chemistry in Biology and Drug Development. It is the week 4 live session, and today we will discuss the some problems which are based on the group week 4 class materials. We will wait for a couple of minutes, then we will start today's session. Okay, so very good evening to all of the students who have joined today's session. Yeah, my name is Moshumi Dev and I am a PMRF fellow of the Department of Chemistry and Chemical Biology from IIT ISM Dhanbad. So today is the week 4 live session is for this NPTEL course of Organic Chemistry in Biology and Drug Development. Today we will solve some 11 to 12 question based on the week 4 class materials, and after solving the questions, if you have any doubt, anything, please do write in the chat box, and I will share this PPT and the record of today's class with the students at the end of today's session. So basically we will solve some MCQs, and these MCQ solvings, this problem solving will help you to do your assignments which is due for the week 4. We will just wait for 2 more minutes and we will start then our session. Let some more students join. Okay. I hope my voice is clear and audible and my slides are visible. So please write in the chat box if you can hear my voice properly and you can see the PPT. So somebody please write in the chat box if my voice is clear and my slides are visible. So my voice is clear and everybody can see my presentation. Okay, thank you so much. Okay, thank you. So again, good evening to the students who have joined today's session. My name is Moshumi Dev and I am a PMRF fellow from the Department of Chemistry and Chemical Biology of IIT ISM Dhanbad, and it is the week 4 problem solving live session for the NPTEL course of Organic Chemistry in Biology and Drug Development, and we will solve some 12 questions today based on the week 4 material. So let's start today's class. If anybody have any question at any point of the today's session, please do write in the chat box, and at the end of the session I will answer those queries. And if still you have some doubts, at the end of the session I will share my email ID. You can mail me anytime about any doubt of this today's class, and I will surely answer you. So let's start today's session. So the first question is the second. Yeah. So the first question of today's session is which one of the following statement is false? The options are as such: both receptors and enzymes have active site. Only enzymes have active site, not the receptors. After binding, reaction takes place in enzymes, and after binding, no reaction takes place in receptors. So when doing this kind of questions, please do read the question carefully for that what it is asking. Is it asking for the false statement or true statement? Because I have seen that many students do mistakes in this kind of question, like this true and false questions. So please read the question carefully before answering this kind of question. I will show you the answer first, then we will discuss this question. So the answer is B, only enzyme have active site, not the receptors. Now we all know that the enzymes, they have a particular site where the substrates bind and the reaction takes place and the products are formed. These are called the active site of the enzymes, where the enzyme generally reacts. So there are some molecules which are called receptors. These receptors do not have the active sites. So what is the difference between enzymes and receptors? In receptors, there will be an area which has a correct shape to accept the incoming messenger. It has the site for the incoming messenger, not for the substrate molecule. Okay. Now what is this messenger? Messenger does, the chemical messenger does not undergo a chemical reaction. We all know that when the substrate binds to an enzyme, there happens a chemical reaction, and through that chemical reaction the products are forming. But the chemical messengers are some molecules which do not undergo the chemical reaction, but it feeds into the binding site of the receptor protein and passes on its message and then lives unchanged. So that means that it is this area which has the reaction, it is the false statement, right? Because only enzymes does not have the active site. Receptors also do have the active site, but no reaction takes place in that active site. It only helps to bind the chemical messenger, and the chemical messenger, after passing the message, it leaves the site unchanged. So this is the false statement among these four, because the first option, both the receptors and the enzymes have active site, it is correct. Okay. The second option, only enzymes have active site, not the receptor, active site, not the receptor, it is false, because both of them have active sites. After binding, reaction takes place in enzyme, which is also true, and after binding, no reaction takes place in receptors, it is also true, because reaction does not take place in the receptors, only the chemical messenger binds to it and it passes on a chemical message. So among these four options, this option B is the false statement. Okay. So I hope I am clear about this question. If you have any doubt regarding this, please write in the chat box. At the end of the session, I will address that doubt. So moving toward the next question. Okay. The problem 2 is, what is the purpose of having two aspartate residues in the aspartyl protease? Option A, one aspartate helps the release of the hydrogen ion from water, make hydroxide ion, and the other aspartate in turn gives the stability to the oxyanion that is being generated. Option B is, two aspartate residues make the active site more flexible. Option C is none of options A and option B. And option D is both option A and B are correct. So aspartyl protease is one type of protease in the 4 class of proteases you have read in this week. So this aspartyl protease, in the active site there are two aspartate residues. So the question is asking for the role, why there are two aspartate residues. What is the role of having two aspartate residues? Okay. So mainly the options are two, you have to just guess the correct option, and first I will show you the correct answer, and then we will discuss the active site mechanism for the aspartyl protease. Okay. So the correct answer of this question is option A. One aspartate helps the release of hydrogen ion from water, make hydroxide ion, and the other aspartate in turn gives stability to the oxyanion that is being generated. So somebody wants to join, so let me just admit. Okay, so now let's just discuss the active site mechanism for the aspartyl protease. So since the correct option for this question, that the purpose of having two aspartate residues is that one aspartate helps the release of hydrogen ion from water, make hydroxide ion, and the other aspartate in turn gives stability to the oxyanion that is being generated. So this is the active site of the aspartyl protease. There you can see that there are, let me just generate the pen. So there are one aspartate residue here and another aspartate residue is here, and in between there are other various amino acids, because it is a protein molecule, you know, there are peptide bonds and there are many other amino acids. So there is a water molecule at the active site of this protease, and there is another peptide bonds is present here. So what is the role of this protease? This protease generally cleaves this peptide bond. Okay. So how does it work? So basically the one aspartate and aspartate ion, it will take the hydrogen ion from this water and make it OH minus. Okay. Now we all know that OH minus is a very good nucleophile, and this nucleophile basically attacks this carbonyl carbon, which is a good electrophile, and this kind of tetrahedral intermediate is generated. Now what happens? This negative charge comes back here, and this bond is cleaved, and in between this oxyanion, this O minus, it is hydrogen, it is stabilized by hydrogen bonding with this aspartate residue. Okay. So what happens? That this peptide bonds is cleaved. So what is the this is the role of having two aspartate molecules. One aspartate this aspartate residue, it is just making water a good nucleophile, so that it can attack the electrophilic carbonyl carbon of this peptide chain, and it can cleave that peptide bond, and we have this cleavage, and we have this acid part and this amine part. And the other aspartate, what it does, it is giving stability through hydrogen bonding to this tetrahedral intermediate. Okay. So basically just remember very brief detail about this active site mechanism. You do not need to memorize all this thing, just have a very clear thought about this kind of mechanism. Okay. So there are all four proteases. It is good if you can I think just understand the active site mechanism of both the four residues, because so it will be easier for you to answer this type of questions. So if you have any doubt regarding this mechanism or this protease, you can write in the chat box. At the end of the session I will answer that. And so let's move toward the next question. Okay. The next question is about another protease, which is the thiol protease. Please read all four classes of proteases very carefully, because this mechanism somewhat it seemed to be little bit of tricky and difficult to memorize, but after understanding the active site mechanism you can see that it is very easy. Just you need to understand what is happening there. Okay, you do not need to memorize everything. So the question is, which of the following statement is true regarding the thiol proteases? Again I am saying, while answering this kind of true false question, please read the question very carefully what it is asking for. It is asking for true or the false statement. Okay. So here it is asking which of the following statements is true regarding the thiol protease, and we have four options here. Option A, it can react only via catalytic triad of aspartate, histidine and cysteine. Option B is histidine is not necessary in this protease. Option C is aspartate is the most important in this catalytic triad, and option D is histidine is sufficient to activate these proteases because of more nucleophilicity of sulfur. So let me just show you the answer first, then we will discuss the mechanism of this thiol protease. Yeah. So the answer is histidine is sufficient to activate these proteases because of more nucleophilicity of sulfur. So before going to this mechanism, I will briefly just discuss every option and let you know why it is incorrect. So the first option says that it can react only via catalytic triad of aspartate, histidine and cysteine. So basically it is false, because for thiol proteases having aspartate is not necessary, okay, and the reason is given in the option D, which is the true option, that histidine is sufficient here to activate these proteases because of the more nucleophilicity of sulfur, okay. And option B is histidine is not necessary in this protease, which is wrong, because histidine is necessary, aspartate is not necessary. And option C is aspartate is the most important in this catalytic triad, which is also false. Actually most important is not I think aspartate, it is the cysteine and histidine are more important. Aspartate, it does not matter if aspartate is present or not, it may in some thiol protease it has aspartate, some thiol protease aspartate is missing. So it can act via the catalytic triad of this aspartate, histidine and cysteine, or it can react also by the catalytic diad, because it may only have histidine and cysteine, but not the aspartate. So in the thiol proteases which have only this histidine and cysteine, they are called react through the catalytic diad. So this is the mechanism of the thiol protease. Again, like aspartyl protease, thiol protease also does what does it do? That it cleaves the peptide bond. So it will cleave the peptide bonds here and make it acid and amine, okay. So it is this thiol group, it is present in the cysteine, and this is the imidazole ring of the histidine. So what happens, that in the since nucleophilicity of the sulfur is more than the oxygen, so the imidazole ring of this histidine is sufficient to deprotonate this thiol group and make it S minus, which in turn acts as a nucleophile and attacks to the electrophilic carbonyl carbon of this amide linkage. And this after doing this electronucleophilic attack, what happens, that this O minus it returns and this amine bond is cleaved. This amide bond is cleaved and it takes the hydrogen from this imidazolium ion, and what happens, there we have this amine moiety here, and this we have a this is a basically this is a thioamide kind of bond, okay. Now what happens, that there is a water molecule present in the active site and it will hydrolyze this kind of amide, thioamide, and we will have our protease, this cysteine back, and the histidine back in the normal state, and we will have a acid. So this is very easy mechanism if you just need to understand the mechanism. If you try to memorize this kind of things, it will be totally difficult because you will forget. So I will always suggest just read the active site mechanism once or twice very carefully, because it is not difficult, it is very easy, and if you understand it, it will be very easier for you to answer this kind of question. So I hope I am clear about this mechanism and this question. So if you have doubt, you always can ask me, and now we will just move toward our next question, okay. Okay. So the next problem of today's session is, which inhibition study deals with the decrease of the value in the both Vmax and the Km with increase in the inhibitor concentration? Option A is competitive, option B is non-competitive, option C is uncompetitive, and option D is suicide. So I think we all know what is the Vmax and what is the Km. It is basically the Kmax is the Michaelis-Menten constant from this Michaelis-Menten equation for the enzymes, and the Vmax is the substrate Vmax is the reaction rate, basically it gives the maximum reaction rate of the enzyme reaction. And to know this kind of studies, it is always good to remember the graphs of this kind of mechanisms, okay. So first we will I will show you the answer, then I will discuss the graphs for this question, and somebody wants to join. Okay. So first I will show you the answer is uncompetitive inhibition. So okay, so these are the Lineweaver Bark plots of the inhibition. So as you can see, to answer this kind of question, it is always good to understand and remember the graphs of how the graphs of these kind of inhibitions looks like. So for first one is the competitive inhibition. So here you can see, so if you remember about that this kind of Lineweaver work plots, the slope of this graph is given by Km by Vmax. Okay, so the uh, slope is given by Km is the Michaelis-Menten constant, by Vmax, and the intercept, it is intercept, I am written as int, intercept is given as 1 by Vmax. In the x axis we have 1 by V, in the y axis we have 1 by S. Okay. So, what the question, it is asking that this red line, it represents the presence of inhibitor, and the blue line, it is mentioning that there is no inhibitor present. So, in the first graph of this competitive inhibition, as you can see that these both curves are meeting at the same point on the y axis, which means that the value of the intercept for the competitive inhibition in presence of inhibitor or no inhibitor, it is basically the same. Since the value of intercept is same, what it implies that the Vmax, it is unaffected, it is unchanged. Okay. Now, but look at the slopes of this reaction. Okay. Since you can see that with presence of the inhibitor, the slope of this red line, it is more than the blue line, means in presence of inhibitor the value of Km by Vmax, it is increasing, but the Vmax is unchanged. So, what it implies? It implies that the value of Km, it is increasing. Okay. Now, let us see the uncompetitive inhibition. So, here what we can see that there are the different values of now there are different values of the intercepts. So, you can see that the value of here the in presence of inhibitor, the value of 1 by Vmax is more than the value of 1 by Vmax when there is no inhibitor, which implies that the Vmax value is reduced. Since 1 by Vmax, this value when there is inhibitor, okay, I am representing as like this, I means inhibitor, it is greater than 1 by Vmax when there is no inhibitor, I mean written as Ni means no inhibitor. Since the value of 1 by Vmax of inhibitor is greater, so what it implies that Vmax value, it is reducing, just you have to just have the reciprocal, means Vmax value, it will be decreasing in that case, Vmax is reduced. So, what happens? Now, look at the slopes of this thing, that the slope of this both of this graph, they have same slope. Okay, the slopes are unchanged. Since the slopes are unchanged and the value of Vmax is reducing, so that mean what it means? To have the same slope as this one, the Km also has to be reduced, so that the Km by Vmax ratio remains same. So, I am again repeating it, that the Vmax is reducing, but the slope is same with inhibitor or without inhibitor, the slope of these lines is same. Since the Vmax is reducing, it is reduced in the with presence of inhibitor, the Km value, it also has to be reduced, so that the ratio remains same. So, just I am giving you an example, let us say if the Vmax for the inhibitor value, it is 100, okay, and the Vmax value for the no inhibitor, it is 500. The Km, so the 500, okay. So, what happens that the slope value, it is 100, okay, let us say not 500, I am just making it, let us make it, I will make it 1000, so that it is easier to understand. So, with no inhibitor, let us say it is 1000, and with inhibitor it is 100. So, since the Km Vmax values are slow, we have to have this kind of relationships. So, let us say the Km value for with this is 100, okay. So, the what is the value of the slope? It will be 1 by 10. So, to be this left hand side and right hand side to be equal, so what the value of this we need to have 10 here, okay. So, we will have this left hand side and right hand side will be same. So, to have this same slope, we have to have a reduced Km value. And in case of non-competitive inhibitor, as you can see that the Vmax values are different here, okay, but here they will meet at some point, so that the Km is unaffected. And similarly, how we did that, in this uncompetitive inhibition, the Vmax value now has to be reduced. Okay. So, the Vmax value will be reduced, which we can determine from this intercept value also. So, this is the three type of graph of the three type of inhibitors. So, the question asks that which of the study deals with the decrease of values of both Vmax and Km. So, the correct answer will be uncompetitive inhibition. Just remember how the graphs look like for this three kind of inhibitor. So, you can answer this kind of questions easily. Now, moving toward the next question, it says that which of the following inhibition is called mechanism based enzyme inhibition. The options are competitive inhibition, non-competitive inhibition, suicide inhibition, and D is active site directed irreversible inhibition. So, let me just show you the answer, and then we will discuss what is the correct answer and why it is called mechanism based enzyme inhibition. So, the correct answer is suicide inhibition. Now, what is suicide inhibition? Actually, in this case, the inhibitor molecule is not strong enough to inhibit the enzyme completely. So, what does it do? That the inhibitor molecule binds to the enzyme just like the natural substrate. There is a reaction takes place, and the inhibitor molecule then will be transformed to a very reactive molecule. So, there is an inhibitor, it will bind to the enzyme and the reaction takes place, and the reaction will create a very reactive molecule, which is I star I am representing, and it will kill the enzyme. So, this is called suicide inhibition, because basically the enzyme is taking part in a chemical reaction which is killing itself completely, because it is forming a this I star will form a strong covalent bond with this E, and it will basically deactivate the enzyme completely. So, this type of mechanism is called suicide inhibition. And okay. So, there is a brief discussion about suicide inhibition here. So, it is known as mechanism based inactivation, because here the reaction is taking place and the enzyme and inhibitor reaction is going through a mechanism which is very similar to the enzyme and natural substrate. Okay. The inhibitor makes the use of enzyme's own reaction mechanism to inactivate it. That's why it is called mechanism based inactivation. In suicide inhibition, the structural analog is converted to a more effective inhibitor with the help of the enzyme to be inhibited. For example, there are few examples, because in questions you can be asked for the examples of this suicide inhibition. So, there are, for example, ornithine decarboxylase catalyzes the conversion of ornithine to putrescine, which is necessary for the polyamine synthesis. Okay. When the ODC in trypanosoma is inhibited, the multiplication of the parasite is arrested. Therefore, the inhibitors of ODC enzyme such as difluoromethylornithine have been found to be effective against the trypanomyces, which is the sleeping sickness. The similar mechanism is observed in the case of allopurinol, which is oxidized by xanthine oxidase to alloxanthine, which is strong inhibitor of xanthine oxidase. The anti-inflammatory action of aspirin is also based on the suicide inhibition. Arachidonic acid is converted to prostaglandin by the enzyme cyclooxygenase. Aspirin acetylates a serine residue in the active center of cyclooxygenase, thus prostaglandin synthesis is inhibited. So, the inflammation subsides. So, these are some examples of the suicide mechanism. You can you can remember, or you may not remember. So, just you can it is good to know some examples. So, I think I am clear about the suicide inhibition and why it is called the suicide inhibition. And if you have doubts, please write in the chat box. At the end of the session, I will address that doubt. So, next moving toward the next problem. Okay. So, which of the following statement is false regarding the ASDII? So, this ASDII, it stands for active site directed irreversible inhibition. So, the options are A, TPCK is an inhibitor of chymotrypsin, B, TLCK is the inhibitor of chymotrypsin, option C, the inhibitors do not have any directional property, option C, the reactive functionality of inhibitor is responsible for this kind of mechanism. So again this is a true false kind of question, so please read the question carefully why what it is asking. It is asking for the false option. So let me just show you the answer first, then we will discuss about this question. Yeah, so the answer is the inhibitors do not have any directional property. Actually it is very easier question regarding, because from the name itself, the active site directed in irreversible inhibitor, the name itself suggesting that the inhibitors do have a directional property. So the option C is 100% incorrect, because it is saying that the inhibitors do not have any directional property, but the name itself suggesting it that active site directed irreversible inhibitor. So option A is TPCK is a inhibitor of chymotrypsin, which is true, and it is you can see it in this figure. I have just given you an example of active site mapping via irreversible inhibitor. And option C is that TLCK is an inhibitor of the trypsin. So it is actually you just remember this what is the inhibitor of which enzyme, just remember the names, it is sufficient. And the option C is incorrect, which I have said. And option D is the reactive functionality of the inhibitor is responsible for this kind of mechanism. So I can explain this option D via this figure. So you can say it is the TPCK inhibitor of the chymotrypsin. Okay. So the TPCK name, it denotes the tosyl L-phenylalanine chloromethyl ketone, and the TLCK it stands for tosyl L-lysine chloromethyl ketone, which structure is similar. So but there is not there is a phenylalanine here. So in the TLCK inhibitor of trypsin, there will be a lysine residue here. Okay. So this is the natural substrate for chymotrypsin. We know what chymotrypsin does. It basically hydrolyzes the peptide bond which is just at the C side of the aromatic amino acid. Okay. So you can see that this is the inhibitor of TPCK inhibitor, which very much looks like the natural substrate for chymotrypsin, but there is a reactive functionality. This is the reactive functionality which is the main reason for this kind of inhibition. This C double bond O CH2Cl, this group is responsible for the inactivation of this enzyme and this inhibitor. And you can see that in the chymotrypsin there is a histidine 57, and it also reacts via a catalytic triad of aspartate histidine, and there is no serine present here. Okay, so in this case the histidine molecule, it has a imidazole ring, and this imidazole ring, this nitrogen, it binds tightly with this this CH2COR group of this inhibitor. Since it is losing this proton, and this proton is very necessary for the activation of this chymotrypsin for the peptide bond cleavage. Okay. But when there is TPCK, it is forming a covalent bond with this inhibitor, and there is no proton present in this histidine. So it cannot take part in the proton relay mechanism for the chymotrypsin. Okay. So this is the reactive group. Now you can see that there is a C double bond O present, there is no CN bond present here, and it is very strong bond. Okay. So that is why it is not easy to break this kind of inhibition. So this is called the active site directed in enzyme inhibition. So I will suggest just read the mechanism for how the chymotrypsin is works, and what is the role of the catalytic triad here so you can easily answer this kind of question. There is serine present here in presence of cysteine. Okay, there is a catalytic triad of aspartate, histidine and serine, and the TPCK is the inhibitor for this chymotrypsin, and this is when we use this kind of inhibitor, the enzymes is got deactivated, and this is called the active site directed in irreversible inhibition, because this reactive functionality, it is taking part in the active site of the enzyme. Okay. So I think I am clear about this. So let's moving toward the next question. Again I am saying, if you have any doubt, please write in the chat box, and if you have doubt after today's class, you can write a mail to me or ask the question in the discussion forum. I will surely answer that question. So the next problem, it is says that which of the following statement is true regarding to the antibodies. Option A, T cells are associated with humoral immunity, B cells are associated with the cellular immunity, B lymphocytes are present in bone marrow. B lymphocytes are generated by thymus. So, this is basically this kind of true false question. Okay, somebody wants to join. So, this kind of true false question you can easily answer if you can know the what is the B cells and what is the T cell, and the comparison between these two kind of cell, and the correct answer of this question is D. The sorry, it will be there is a mistake, typographical error, it will be C here. Okay, I am just going to correct it right now so I can update it later. So, the correct option is C, the B lymphocytes are present, the B lymphocytes are present in the bone marrow. Now, what is the B cells and what are the B cells and what are the T cells? So, when there is a attack to our body by the foreign particles, which are the inhibitors like bacteria, there can be some which are called the antigens, basically the foreign particles. So, there are two kind of cells in our immunity system. First one is called the B cells, and what the B cells does, it directly attacks the inhibitors outside the cells. Now, when the invaders are attacking our body, it can attack our cells directly, or it can be they are present in the bodily fluids, okay, before attacking the cells. So, the B cells are taking care of our immune system by attacking this inhibitor outside the cells in the body fluid, and the T cells, what it does, it attacks the infected cells. So, that is a basic difference between B cells and T cell, which is showing by this cartoon very well, that the B cells, what it does, that it attacks the inhibitors outside the cells, and T cell, what it does, that it attacks the infected cells directly. So, the first two options we can easily say that these are false, because the T cells are not associated with the humoral immunity and the B cells are not associated with the cellular immunity. Okay, this will be just otherwise. Okay, the T cells basically are associated with the cellular immunity and the B cells are associated with the humoral immunity. Okay, B lymphocytes are present in bone marrow, which is correct, and B lymphocyte generated by thymus, it is wrong. Actually, T cells are generated by thymus, and it is very easy to remember because T stand for thymus and B stand for bone marrow. So, you can remember it like this. So, this is basically the differences between the B lymphocytes and T lymphocytes, which are also called the B cells and T cells. The first one is that the B cells, which origination and maturation takes place in the bone marrow, and for the T cells, the origination in bone marrow, but it is generated by thymus. Okay, the maturation takes place in thymus, but it is originated in the bone marrow. So, after all, it is not present in bone marrow, it is originated, but it is present in thymus. But the B cells, their origination and maturation both takes place in the bone marrow. Okay, the B lymphocyte, they differentiate into plasma cell, and the T lymphocyte, they can differentiate into the plasma cell or memory cell or cytotoxic T cells. The B cells are involved in the humoral immunity and the T cells are involved in the cell mediated immunity or cellular immunity, and the B cells, they produce these antibodies, and the T cells, they help B lymphocyte to produce antibodies. So, the T cells does not produce antibodies itself, it helps the B cells so that the B cell can produce antibody which can bind to the antigen and deactivate that antigen, that foreign particle. So, I think it is very clear question, just you have to remember the comparison between the B cell and T cell to answer this kind of question. Yeah. So, the next problem is, it is called which of the following is known as antigenic determinant. Now, the antigens, the antigens are the foreign particles which are entering our body, and there is something in that antigen which can be determined by the antibodies which are generated in our body by B cells. So, we know that antigen antibody, this interaction is responsible for the immune response in our body when we are infected with some foreign particles, some bacteria or other things. So, what is the antigenic known as antigenic determinant? The options are hapten, option A, option B is epitope, option C is enzymes, and option D is paratope. So, let me just show you the answer first, then I will discuss each of these four, so you can understand what is called the antigenic determinant and why. So, I think somebody wants to answer. Okay, if you have any question, please just write in the chat box, so it will be easier for me to monitor that question. Okay, so the answer of this question is the epitope. Now let me just discuss what are the haptens. So haptens, a hapten is a molecule too small to stimulate antibody formation by itself. So it is not associated with the antigen, it is associated with the antibody. Okay. However, when the hapten is combined with a larger carrier molecule, usually a serum protein, the hapten and its carrier together form a conjugate that can stimulate an immune response. So, this hapten molecules, they actually conjugates with a carrier molecule, which is the serum protein, and it can generate the antibody response, immune response. The enzymes, an enzyme from the antibody and enzyme, it is often called a catalytic antibody, is a monoclonal antibody with catalytic activity. So, enzymes, it also is not associated with the antigen. Okay, it is associated with the antibody. Basically, it is a antibody which has a catalytic activity. Now, epitope. An epitope, also known as antigenic determinant, is a part of an antigen that is recognized by the immune system, specifically antibodies, B cells or T cells. And the paratope, it is a part of an antibody which recognizes the antigen. The antigen binding site of the, that is the antigen binding site of the antibody. It is the small region of 15 to 22 amino acids of the antibodies FV region, and contains part of antibodies heavy and light chains. So, in this figure you can easily say it is a antigen molecule. Okay, this antigen has some binding sites which is called the epitope, and this epitope is this part of this antigen, it is recognized by this paratope, this junction, it is called the paratope, which is associated with this antibody. And as you can see from this is a antigen antibody complex, and this antigen antibody complex is basically responsible for the immune response, which is shown by our immunity system. So, this epitope, it is known as antigenic determinant, and other three options, this haptens and enzymes and paratope, it is associated with the antibody. Okay. So, I think it is you can just it is you have understand this question, that what is the epitope, what is the hapten, and what is the paratope and enzymes. So, just remember that the epitope is the is part of an antigen which can just interact with the paratope, which is the site of an antibody, and this epitope paratope, this kind of interactions is responsible for our immune system, immunity response. So, if you have any doubt, please write in the chat box. At the end of the session I will just discuss that doubt. Okay. So, moving toward our next problem. So, what is the use of hybridoma technology? The option A is separation and isolation of monoclonal antibody from a polyclonal set. Option B, separation and isolation of lymphocytes from immunoglobulins. Option C, separation and isolation of haptens from epitones. And option D is separation and isolation of haptens from enzymes. So, I will just show you the answer, and then we will briefly discuss what is the hybridoma technology. The answer is separation and isolation of monoclonal antibody from a polyclonal set. So, let us discuss about this, what is the monoclonal antibody and what is polyclonal antibody, and then we will discuss what is hybridoma cells and what is hybridoma technology. So, basically monoclonal antibody, it reacts with a specific epitope on a given antigen. So, as you can see that there is a actually in the antigen there can be several epitopes. Okay, if there is a antigen, it can have several epitopes on it. Okay, and the antibody, it has which have the paratope, it can bind with the various epitopes of the antigen, or with the one single specific epitope of the antigen. If the antibody it's binding to a specific epitope of the antigen, it is called that monoclonal antibody, and if it reacts with various epitopes on a given antigen, it will called a polyclonal antibody. So, this is the difference between monoclonal and polyclonal antibody. So, in a polyclonal set of antibodies, there are several monoclonal antibodies, and to separate and isolate this monoclonal antibody from this polyclonal set, this hybridoma technology is used. Now I will show you a figure about the hybridoma technology first. Yeah. So what is hybridoma technology? The hybridoma technology, the names coming from hybridoma cells. So what happens that we know the B cells, they are producing the antibodies. Okay. So but these B cells and these are very short lived. To separate this polyclonal from this polyclonal set to separate monoclonal antibody, we need sufficient time. But since the B cells which are producing these antibodies, these are very short lived. So what we need to do? We need to increase the lifetime of these B cells. Now how can we increase the lifetime of this B cell? So there are some cells which we all very are familiar with, which are called cancer cells or myeloma cells. So these cancer cells, they basically are known to be immortal because they never stop the reproducing itself. Okay, it just keep reproducing the more cells, and that's why they are called malignant. So these malignant cells, they myeloma cells. So what does it do? This technology do? They will take the B cells. Okay, the B cells from they will actually they will this in this technology, the mice it is given the antigens, and after the production of the B cells, they are isolated and they are fused with the myeloma cancer cells. Okay, and this fusion of this B lymphocytes and myeloma, they will basically give us cells which is called the hybridoma cells. Hybridoma cells. So hybridoma cells is basically a hybrid cells of the B cells and the myeloma cells. Now these cells are they are basically cultured in the HAT media. It is a medium for the cell. Okay, somebody, I think this is the medium where the cells are actually cultured. So there are many actually culture mediums, and for different mediums are used for different purpose. In this purpose, this HAT media is used. It is good to remember this name also, this HAT media, which is used for the culture of the hybridoma cells. And then each screen, the cells are actually screened, and then after propagation of the positive clones, the monoclonal antibodies are separated from the polyclonal cell. So just you don't need to remember each and everything about this hybridoma technology. Just remember why it is used. It is used for the separation of the monoclonal antibody from a polyclonal set. And what does it do? It basically is the fusion of a B cells with the cancer cells, and the fusion of these cells, it's called hybridoma cells, and these hybridoma cells are cultured, and after the propagation of the cell propagation in the culture medium, the in vivo and in vitro the culturation are done, and the antibodies are separated. You don't need to remember the each and every detail of the technology. Just remember why it is used and what is hybridoma cells, and what is monoclonal antibody and what is polyclonal antibody. Okay. So I will just stop this question here, and I will now move toward the next problem. So it is basically a mathematical problem. So what is the ratio of the V0 by Vmax when the substrate concentration equal to 7 km? The options are as 7 by 8, 1 by 7, 5 by 7 and 8 by 7. Since this week 4 module, it contains various enzyme inhibition and some enzyme kinetics, we will have next 3 questions. We will have some basically numerical questions, so that you can answer the numerical problems of your assignment. So this first numerical question, it says that what is the ratio of V0 by Vmax when the substrate concentration equal to 7 km. Let me show you the answer first, then I will show how it is done. So it is basically very easy. So you just need to remember what is the Michaelis-Menten equation. So it is just V0 equal to Vmax into substrate concentration divided by Km plus substrate concentration. V0 is the initial velocity, which is in the moles per time unit. Substrate concentration, which is in molar unit. Vmax is the maximum velocity, means the maximum for the enzyme work, the maximum velocity. And the Km is the substrate concentration at half Vmax, which is also known as Michaelis-Menten constant. Okay. So basically this is the solve of this question. So what happens if you just take this Vmax in this left hand side? So we will have this V0 by Vmax, and it is equal to substrate concentration divided by Km plus substrate concentration. Now, in our question it is given that the substrate concentration, it is 7 times of Km. So, we can replace this substrate concentration by 7 Km in our equation. So, what it will happen, that just replacing this substrate concentration by 7 Km, it will give us 7 Km divided by Km plus 7 Km, means 7 Km divided by 8 Km. So, this Km cancel out and we have 7 by 8. So, it is basically very easy question, easy numerical, but just you need to remember this Michaelis-Menten equation very very by heart. Just remember this equation by heart, and most of the numerical problems will be easily solved if you remember what is this Michaelis-Menten equation and what the these things stand for. So, V0 is the initial velocity, Vmax is the maximum velocity, S is the substrate concentration, and Km is the Michaelis-Menten concentration. So, I think this question was very easy, and you can easily solve if there is in our assignment, if there you get this kind of questions, you can easily solve. So, just remember Michaelis-Menten equation and the graph by heart. So, the next problem, I will quickly move to the next problem. It says that consider the following plot of velocity versus substrate concentration of the enzyme catalyzed reaction obeying Michaelis-Menten kinetics with an initial enzyme concentration of 10 to the power minus 5 micromolar. The turnover number of this reaction is given by these numbers. So, okay, I just missed the graph. I will. So, this is the graph. Okay, by in the y axis we have V, the velocity of the reaction in micromole per minute unit, and in the x axis we have substrate concentration in 10 to the power 3 molar, and you can see there is a this point. Okay, then so this value, it is called the Vmax, the maximum velocity. Okay. Now, the question, it is given that the initial enzyme concentration is 10 to the power minus 5 micromolar. Okay. And I will show you the answer first, then we will solve this question. Okay, the units is unit of this turnover number is minute inverse. So, minute inverse, I will show you the solve. Okay. So, as I said that what is let me first say what is the turnover number. So, basically the turnover number of the enzyme, it represents the number of molecules converted to products in unit time by one molecule of enzyme. Okay, you can see it here that K2, this turnover number of an enzyme, it represents. Okay, let me just use the highlighter instead, if it will be easier for you to see it. Okay. So, turnover number of an enzyme, it represents yeah, it is. Just remember you should know what is the turnover number of the enzyme, because it is very important if you have to just compare various enzymes on the basis of its activity. It is done on the basis of the turnover number of an enzyme. The more the turnover number of the enzyme, it is activity is more. Okay. So, the turnover number, it is basically the number of enzyme, it represent number of enzyme, it represent that the number of molecules converted to products in unit time by one molecule of enzyme. It is called the turnover number. So, again I said that remember Michaelis-Menten equation by heart, because most of the numericals will be solved itself easily if you remember this Michaelis-Menten equation. Okay. So, it is that the equation which in the previous slide I have shown you, that it is that in the previous slide which I have shown you, it was okay, just let me move toward the previous slide. It is the Michaelis-Menten equation, and there is another form of this equation. Sorry. So, this is the V0 equal to K2 S0 and E0 divided by S0 by Km. Now, what is S0? It is the initial substrate concentration. Okay. The initial substrate concentration is called S0, and E0 is the initial enzyme concentration. So, now at the just beginning of the reaction, okay, when the not at the beginning of at the high substrate concentration, as you can see this way the reaction is going, the graph is going. At this point, this is the highest substrate concentration, and at this point the velocity is just constant, it is zero order reaction. Okay, it does not increasing anymore. So this point of velocity is called the Vmax, and it is happening when the substrate concentration is high. So we can say that at high substrate concentration the value of S0 which will be much greater than Km. And Km, what is the Km? The Km I have shown you in the previous slide. It is Michaelis-Menten concentration, which represent the substrate concentration at the half Vmax. So at the higher substrate concentration, when the value is much greater than Km, in the denominator of this equation we can just in the denominator we can just skip this Km part, since S0 is very much greater than, then actually what happens that S0 plus Km value in the denominator, it will be roughly equal to S0, like it is just adding adding 10,000 with 0.1. if you can just take this as an analogy. So we can say it is 10,000, roughly we can say that the S0 will equal to S0 by plus Km. So since the denominator of the Km is vanishes, what happens, we only have this S0, and this S0 and S0 of the numerator, it will be basically cancelled out. Then what we will have, the rate equation we will have like this, that we have K2 E0, only K2 and E0, because S0 and S0 is cancelling out, and it is representing the Vmax. Vmax is the rate attains at the maximum value, and the reaction is, it is zero order now with respect to the substrate concentration. So now according to our question, the values we are given is the E0 value, the initial enzyme concentration value is given 10 to the power minus 5 micromolar. Okay. So and the Vmax value from this graph we are having 8, and the unit is micromole per minute. Okay. So it is so what is the value of the turnover number? The K2 value, K2 is called the turnover number. This K2 the K2 is called the turnover number. So what is the turnover number from this equation? So the K2 equal to K2 E0 equal to Vmax, and the K2 value is Vmax by E0, and the Vmax is given as from the graph is 8 micromole per minute, and this or the 8 micromole minute inverse, and the E0 value is given as 10 to the power minus 5. So after doing this mathematics, there is a typographical error here. So just it will not be 10 minus 5, it will be 10 to the power minus 5. Let me just correct it here, otherwise when I will just write this PPT, you will just get confused. Okay. Okay. So just remember, it is just it will be superscript value. Okay, this it will be 10 to the power minus 5. Okay, I will go to the presentation mode. So the answer is this. So I think I am clear about this mathematics. Just remember what is the turnover number, and how this Michaelis-Menten concentration is reducing to this form. And after having just looking at the graph carefully, you can know the value of Vmax, and the initial enzyme concentration is given as 10 to the power minus 5, and by this you can easily do this math. So I hope I am clear about this math, and if you have any doubt, please write in the chat box. And the last question of today's session, it is also a numerical. So it is saying that consider the following plot of 1 by V0 and 1 by S plot in the Lindweber-Bark, and the results are obtained from this graph is the intercept value is 0.1 micromole per minute inverse, and the slope is 5 into 10 to the power 3 minute. So what is the Km value for the reaction? So these are the options. So I will show you the answer, and we will solve this question together. And this is the Lindweber-Bark plot. As you is this it is this kind of questions from the graph are very easy to answer if you just remember the specific values, that in the y axis we are plotting 1 by V, in the x axis we are plotting 1 by S, and the slope of this line, it is Km by Vmax, and the value of this intercept, it is 1 by Vmax. So in the question what we are given, the intercept value are given, means 1 by Vmax value is given, and the slope is also given, I mean the Km by Vmax value is also given. So it is very easy to have to know what is the value of this Km, and the answer is this. So let's just I will just let you know what is the slope and what is the is. So the equation of the Lin-Weaver-Bark plot is like this: 1 by V0 equal to 1 by Vmax plus Km by Vmax into 1 by S. So just compare it with the normal equation which we all know is y equal to m x plus c, or I will just write it otherwise, it will be easier. So, or we can write it c plus mx. So, y is the value at the y axis, x is the value at the x axis, and m is called the slope and c is called the intercept. Just compare this equation with this one. So, you can see at y axis we are plotting 1 by V0, at the x axis we are plotting 1 by S. So, the intercept for this equation is 1 by Vmax and the slope is Km by Vmax. So, since we are plotting this and we are having this same thing is written, that intercept and slope, intercept is 1 by Vmax, slope is Km by Vmax. Now, from the equation, from the data given to you is 1 by Vmax is 0.1 micromolar minute inverse, and 1 by, so what will be the Vmax value? The Vmax value will be just the reciprocal, means 10 micromole per minute. Okay. Now, the slope of the slope from the data is given is 5 into 10 to the power 3. So, what will be the value of Km? The value of Km will be slope multiplied by Vmax. So, the value of slope is given by this, the value of Vmax which we calculated is this. Now, the value of Km we are getting as 5 into 10 to the power 4 micromolar. So, again I am just discussing the Lin-Weaver-Bark plot. You can also have another plot is can be given, and another values of the slope and intercept is given, but just remember it is very easy. Just write the equation in the y equal to mx plus c. Just see what is the plotted against y and what is the plotted against x axis, and from that equation you know the value of slope and intercept, and from the values given to you the data, you easily can calculate this values which are asking in the equation. Okay. So, that is it. These were some 12 question of today's TA session, and I think you guys understood more of the questions, most of the question. If you have any doubt, please write in the chat box, I will answer that doubt. And if you have doubt after today's class, I will just share my mail ID here in the chat box, and you can email me at any time if you want. Just show me, just share my email ID with you. So, if you have any doubt, please write in the write me a mail regarding that doubt, or you can also just I will just stop presenting. You can also write the doubts in the forum of the discussion forum of this NPTEL course of use organic chemistry in biology and drug development. And these PPTs, these slides, I will share it, and today's recording of today's session I will also share that recording to you. And if you have still any doubt, please do write a mail to me or write in the discussion forum. And again we have this kind of TA sessions in every Friday from 7 to 8 p.m. regarding that weeks. Today was week 4, and next week at the same time from 7 to 8 p.m. we will have another TA session about the week 5. So, please do join that TA session, and we will discuss next week course material, and this some 11-12 questions there also, and this will help you doing in your assignments. So, thank you for joining today, and I hope I was clear about the question more or less, and I will see you next week. So, thank you so much who have joined today's class, and I will see you the next week, and now I will close today's meeting. Okay, thank you so much.